All right, folks. Honestly, I'm very excited about this. When you say honestly, it means that other times it's not honest. So you gotta be careful when we're doing this. This is very exciting for me because it took a while to arrange this podcast episode. And I'm excited because there's four of us on this call, and we are all wearing the Healthcare Unfiltered t-shirt, which is like probably unprecedented. Yeah, I know. This is a great, this is the best. But in all seriousness, I have three amazing clinicians, faculty, investigators, researchers who want to do the best for our patients with cancer in general and multiple myeloma in particular. And I've invited the three of them because we want to talk about just the complexity of designing and executing on clinical trials. And we're going to start with some introductions, but really the purpose of this podcast is what does it take to do a clinical trial from initiation to completion? What are the problems? What are the hidden spots that we need to look at? And for that, we have junior investigators and a senior investigator with us, because the senior dude is going to tell us about the problems when he was a junior guy. All right. So Hira, I want to start with you. A little bit about you, introduction, where you are, where you practice, and what got you into the field of multiple myeloma and becoming a clinical trialist.
Sure. Thank you so much for the invitation. I'm really thrilled to be here. So I'm Hira. I'm a hematologist and a researcher at McMaster University in Canada. And so I finished my fellowship back in 2016, spent a couple of years doing a master's, and then have been on faculty for the last 4 years. I have a special interest in older adults with multiple myeloma, and so particularly focused on clinical trials around that, and then work with two of our cooperative groups in Canada, the Canadian Myeloma Research Group, as well as the Canadian Clinical Trials Group, and then I'm a board member for Myeloma Canada. So have done all my training in Canada, so hopefully I'll give you a slightly different perspective about challenges that are unique for our patients and how unique in terms of doing clinical trials as well.
And Hira, is there something specific that sparked your interest in older adults or myeloma specifically, or just natural stuff that happened?
I really like the longitudinal patient relationship. So I love seeing the patients at the time of diagnosis and getting to know them now for over a decade. And so for me, part of it is seeing in fellowship, patients that you get to see them longitudinally over time was one of my key reasons. And I do genuinely enjoy taking care of older adults. So it was a natural fit to be in multiple myeloma.
Okay, as long as older is a moving target. I get nervous whenever they say older. Now I'm becoming close to that age that Hira is gonna take care of. Murali, a little bit about you, where you are, where you practice, what you do, and what got you interested also in the field of myeloma.
Sure. So my name is Murali Janakiram, and I'm an assistant professor at City of Hope. It is really a pleasure to participate in this along with Shadi, Hira, and Saad. Thank you for the opportunity, Shadi, for bringing us all together. My interest is in immunotherapies, specifically cellular immunotherapies in multiple myeloma. That's where most of my focus is on. I have been part of a member of the Eastern Cooperative Oncology Group, or ECOG, for quite a few years, and have been accustomed to the group as well. What got me interested in myeloma? Actually, I don't know. That's the real answer. So I started doing myeloma. I just got interested and stayed in it all that.
Sometimes it's interesting. Like I feel, I'm curious to see here Saad's thoughts. I sometimes feel our career is shaped. Some of it is luck, which is the mentors you work with. Honestly, you start your fellowship and you end up, getting paired with a faculty member, and you just like it and you keep going. and sometimes could be interest in the biology of disease. In my case, as an example, was totally completely luck. I was at Northwestern. I started working in malignant hematology, and I liked it. And probably if I was linked with more solid tumor, people would have been different. Saad, how about you? A little bit about you. You've had obviously a longer career than Murali and Hira, but tell us about you, where it started, and where you are now.
No, thank you. It's actually my privilege, actually, Murali and Hira, to be with you guys and share some of my experiences. When I got into my fellowship training, Chari, I actually wanted to be a breast oncologist. So I had done rotation in Sophia Maraver's lab at University of Michigan Ann Arbor, and that's what I wanted to do when I started my fellowship. But I went to this nice seminar on heat shock proteins, with my later lab mentor. And around that time, the unfolded protein response was being looked at, along with proteasome inhibition. So this is like, mid to, 2006, 2007. And that's what got me interested in myeloma. I ended up spending, time in the lab. And because I was on a J1 visa, and really looking for an academic myeloma place, I went to Arkansas as my first faculty position. And the rest is history. I had very strong, although old-fashioned tough mentorship from Dr. Barlogi, but that really propelled my career. And it was a major crash course in clinical and translational science, as well as how to do clinical trials, how to think critically about some of these questions and innovate. So, it's always a pleasure to, chat with the, younger investigators. I don't call them junior investigators, because, you guys will fix this problem.
Saad, I know a couple of things about you that I'm going to say. Number one is you have an MBA. Tell me why you went back to get an MBA.
So, it's different, how your career kind of evolves. And as I was getting more into the mid-career mode, I was recognizing it's so important for you to understand the holistic business of medicine, and it's important for you to appreciate what's really happening in the world. We talk a lot about cost of drugs, but I understand it so much better now in the context of, how the world economy works, and what influences, a lot of those decisions. So, even, the administrative task, how do you, project, the growth of your service or your department? How do you, think about, the money matters? How do you frame the argument to get resources for what you want to do? I think of things in a very different way compared to how I did 5 or 6 years ago.
And you're currently, Saad, at Memorial as the chief of the myeloma service. What does that mean? Like, how many myeloma physicians are there? Obviously, it's we all know how big Memorial is, but how many physicians do take care of myeloma within the Memorial system?
So we have, I have 9 physicians in the myeloma service, who are part of my team, but really the myeloma care team is comprising of 16 people. So we have, people like Sergio and Heather Landau and David Chung, who are part of, adult BMT and transplant cell therapy, but, we're part of the combined myeloma care team. So, in terms of our research meetings, our, multidisciplinary rounds, we do it together. And, I live in 3 services. So I'm part adult BMT, part cellular therapy as well. So, that makes for a very, good multidisciplinary approach when you're thinking about doing clinical trials. We're not thinking individually, we're thinking as a team. So we talk about, okay, where are the gaps? What are the questions we want to ask, and develop our portfolio of research that way.
Before I go back to Hira, I just want to ask you I want to make sure listeners obviously understand the breadth of expertise that we're talking about. Do you know how many clinical trials you have led or co-led and in the myeloma space or in general, including randomized control trials? I just want to make sure at least that listeners realize that you have the expertise to even opine on the topic. Otherwise, why in the world do I have you on?
Was that question, posed to Hira or to me?
No, it was to you. Then I'm going to go to
Okay,
Hira.
all right. Okay, sorry. So now that I'm not keeping counts or numbers, but I would say I've been part of probably close to 50 different clinical trials in my career so far. Whether it's at sub-I or investigator or lead investigator in terms of randomized, phase 2 or 3 studies, probably, 17 or 18 that I've been part of, several that I've led and have published on.
Well, you could definitely do a better job, though. I mean, I don't think I'm not that impressed, but I think you could certainly get on and do a little bit more, please. All right. So here, before I get started, I just want us to agree maybe on a couple of principles, and you tell me if they're completely off. So, are we able to answer every question with a prospective randomized control trial? Hira?
So I would say we would want to, but I don't think that's possible.
Why? Why can't we?
I think there's actually lots of reasons for that. And one of them is that, going back to older adults, for example, older adults sometimes are underrepresented in clinical trials. Clinical trials may not be powered enough for them. So in those cases, actually having registry real-world studies to evaluate outcomes can be equally as important. So not to say that randomized clinical trials are not incredibly important, but when we look at the evidence and how to actually treat patients, I think that, looking at some of these additional methodologies is equally as important. And then lots of trials, and Saad probably knows this, more than anybody else, you can't always, there's so many barriers in leading clinical trials and executing clinical trials. So although we want to be able to answer everything through a randomized clinical trial, that is the ideal, that is just not feasible. So having some alternative ways to still be able to answer those questions is really important.
Murali, what your thoughts? You agree? Any other comments in terms of can we do everything with an RCT, prospective RCT, or we have to be more creative?
Agree with Hira that there is no way we can do prospective RCTs for every question. And one of the difficult, good and bad things about what is happening now is a tremendous pace of science, especially with respect to how fast effective drugs are brought upon. And because effective drugs are brought upon quickly, if we have to do one randomized trial, even for a relapsed refractory disease, it will take more than 10 years, while by that time, we might have 2 or 3 equivalent or better drugs. And we really need to consider how we can bring those to the patients faster. So I would say it is getting tougher and tougher to answer every question through a randomized clinical trial with an endpoints of even PFS, let alone overall survival. That doesn't mean we shouldn't do it, but I think we need to find innovative ways of comparison for answering the same question.
Saad, do you tend to agree with that?
So in terms of doing clinical trials for niche populations, I completely agree. For niche populations where we either don't have a standard of care or we extrapolate a standard of care for those patients, I think doing single arm studies, is a very feasible thing. One of my, mentors, Bob Orlowski, I really like his saying from the early time when I was a young investigator in SWAG. He said, Saad, there are trials that you want to do, and then there are trials that you can do. So I think being a bit pragmatic, is very important for those niche populations. I think, so you don't need randomized trials to answer every question, but you do need randomized trials, to influence the standard of care, especially for, the frontline patients. Endpoints is a totally different discussion, and I'm sure we're going to get into, what would be the appropriate endpoints. But I think at least for the broader, frontline patients, not the niche populations like, the elderly, frail, or plasma cell leukemias, etc. I think randomized studies would be the right way to do it.
So Hira, let me go back to you, and I want to hone in a little bit on your experience and what you, and then I want to go to Murali with that, and then I will have Saad reflect on that experience you've had. So when you decided to design a clinical trial and try to take it forward, what's that process like? How did you decide that I want to attempt to answer which question? Because so many questions out there, right? I mean, there's so many questions. Take me through your mindset, how you decided on the question you wanted to answer, and how you went about it. So then we can maybe reflect on the difficulties and what you went through.
Okay, sure. No, thank you for that question. And I look forward to hearing some of the feedback about where I was wrong or right. So as I mentioned, my interest again has been in older adults with multiple myeloma. I also practice in an entirely publicly funded healthcare system. So some of the questions that I want to know the answer to are actually quite unique. So to give you an example, myeloma currently is 3% of all cancers in the province I practice, yet takes up 20% of the budget. So what I can use for my standard of care is very different. So questions around sequencing, reusing drugs are actually really important. Like, I can only get lenalidomide once. Every drug I can only get once. And if you progress on it, you will never get access to it again. So a lot of the cooperative group with the Canadian Myeloma Research Group, and I work with the Canadian Cancer Trials Group, some of our questions are a little bit more unique that we've asked. So you can look at some of the trials that, again, some of my mentors have led, like Donna Reese, where they're about adding things like cyclophosphamide to different agents, because that's all we have, and we need to maximize every ounce of every regimen that we have. So one of the questions that I've been particularly interested in is about continuous daratumumab, right? And that has a particular role now in older adults because as of February of this year, we finally have access to publicly funded DRD for frontline transplant ineligible patients, which is an incredible opportunity in Canada, but represents one of the most expensive regimens for a patient who's going to be potentially on it for 5-6 years. It also means you'll never get access to another anti-CD38 because you would have progressed on it at any line of treatment.
So for the non-myeloma listeners of the audience, I want to be sensitive to them. Maybe just spell out what's DRT, what is
Sure. So, one of the regimens is daratumumab, lenalidomide, and dexamethasone, which is kind of really as one of the pivotal regimens now for transplant ineligible older adults. It's a fantastic regimen with overall survival benefit, fantastic PFS, but it's unique because it's given continuously. So it comes with a huge treatment burden and financial burden for a publicly funded healthcare system. It also limits options for patients later on because you will not get exposed to that class of agents again. So one of the questions we've been really interested in is could we use some form of a fixed duration anti-CD38 or daratumumab, get the most out of it, make it more feasible in a publicly funded healthcare system, and then also allow patients to reaccess it. So that's really been one of the ideas, and I'm happy to talk about it further, but really one of the ideas that I've been taking forward with a lot of the clinical trials group. And again, because until we had access to that agent, there's no way to ask that question of fixed duration treatment, because as you can imagine, that's not a very pharma-friendly question. And with limited, now it's really hard to do any cooperative group trials. So that's one of the questions we've really been trying to take forward in Canada, which may be a bit more unique for us, but especially for the patients I see, it's an extremely relevant question.
So you had this idea, you like it, you're convinced it helps patients in Canada, which is fair, this is where you practice. What is your next step after you have the idea? Do you call a friend? Do you call somebody like Saad or Vincent Rajkumar and say, hey, is this a crazy idea or is it a good idea? Like, what's your next step?
Okay, so great. So I have a smaller group. Our cooperative groups are not as big. So, we're all kind of on a WhatsApp group. We all get together for drinks. Like it's easier. So,
definitely way more fun than the Americans. Look at this.
We are, the Canadian group is definitely more fun. And so, I've been floating around this idea for a while. We've talked to some pharma companies as well to float different trial design ideas. Overall, there was appetite for this idea. So since that time, I can tell you, we originally came up with this idea. It's probably been about 4 years. Until we had access to the publicly funded drugs, this idea was entirely stalled. We couldn't do it because if we weren't going to have Daratumumab, there's no way one could ever afford that. And since that time, we have now taken it to our the Canadian Cancer Clinical Trials Group, which again, I have lots of great senior mentorship in that. And so we've been slowly moving this along. We've tried lots of partnership with lots of different pharma companies, even for correlative work, but again, everybody has a different way of how to do things. And one of the things I've kind of really learned over the last few years, and Vincent Rajkumar has been involved in this idea for quite some time and has been really helpful in trying to help us move this along, is that, he said to me, he's like, well, you're not going to be able to please everything. Just think, please everyone, but also don't totally come off the table. So meet people halfway, because again, in the Canadian group, I have people like Nizar Bayless who are, very translational focused, and unless it's an MRD endpoint or MRD's incorporated, it can't be done. But I know at the cost of MRD, it won't be feasible. So there's lots of nuances where we really had to come up with an idea that we could please as many people as possible and slowly take it forward. So the current status of this trial is that, we have hopefully approval at the national level. Now I have to seek the funding, which is going to be the absolute biggest barrier for this. When I've talked to Vincent Rajkumar and some of the other folks around this idea and told them the cost of it, it's actually a pretty cheap trial for how expensive Daratumumab is. But different agencies work differently in Canada, right? So the healthcare funding agency, research agencies, there's no crosstalk between them. So they can't see that even if we were to save a year off Daratumumab, it would probably pay for the trial itself. So that's kind of where I'm at with it. I would say I've been very lucky to have lots of mentorship, and I cannot emphasize that point enough. One of the parts I forgot to mention earlier was, why did I pick older adults? It's because I met Tanya Wilds through my career trajectory and through ASH CRTI, and it was the pivotal moment. I wanted to be a community hematologist, never do an ounce of research. And Tanya entirely changed my career trajectory. So one person can influence a lot. And also, when I have small successes, I have people to celebrate. And when I fail, I have equally people who are just as invested. So mentorship has been key, and slowly we're moving along this idea and have gotten lots of support around it. So maybe if you call me back in another few years, I'll tell you whether I was successful or not.
But Hira, so you started this 4 years ago, like the idea came to you 4 years ago. The trial has not even launched yet.
I don't even have it funded.
You don't have it funded. The idea is there, you got it. So it's 4 years and it's not there. Saad, first of all, we all know that Vincent Rajkumar is always wrong. This is always the case. We don't need to, hopefully he's listening and he realizes you disagree with everything he says. But this is 4 years and it's not even launched yet. So, the field is moving fast. How would you approach something like this and a problem like this? And how would you Like, what's your reaction to the experience that Hira has had over the past several years?
Her challenges are unique. Before I comment on that, first off, Vincent is one of my really close friends, and and I consider him one of my mentors. I've learned so much from him about the years, and
but you're still going to disagree with him.
I do disagree with him, and that's the beauty of friendship. You can have different academic views and still be very good friends, because at heart that what you're trying to do is get better answers for your patients, and move the field forward. With that being said, the challenges that Hira is facing are very similar to what we face in the US cooperative group mechanism. When I was, again, a junior investigator in the co-op mechanism, I used to get frustrated. We had good ideas, but then it would take forever to vet those ideas through the various committees, and then try to get funding from or commitment from pharma, and then get the key stakeholders in the NCI CTEP mechanism and the NCI myeloma steering committee to get the trial going. So I'll give you a very nice example here. So this SWOG 1803 study with Dara-Len versus Len maintenance, this idea actually started as an IGZA-Len versus Len maintenance in 2012. Okay, and Jatan Shah was still in academics, and he was the investigator who came up with that idea. And then there was a lot of back and forth, and OS had to be the primary endpoint, and we were MRD couldn't be incorporated because it was too early. There was lack of data. So eventually, by the time that idea was coming to fruition, it was 2016, and Dara had entered into the schema of things. So then we had to go back to the drawing board. Jatan left academia. Then we got Amrita to be the PI, and that idea. So by the time that original idea from 2012 came to fruition, it was 2018. The study started in early 2019, if I remember correctly. But halfway through the study right now, that question is becoming antiquated. Okay, so the challenge with the ideas that we generate is we don't know what that 5 or 6 year horizon is going to look like, because a lot of innovation is not coming from academia, it's coming from pharma. And so that becomes a challenge. So my advice, is always look at that 5 year horizon with your geographic area. So I think the question you're trying to ask is still relevant in Canada, and just pursue it. And we can talk about offline how to think about getting that kind of study off the ground. I'm not sure if Sikandar Alavadi has reached out to you or talked about his trial that he's been trying to do in the older patient population, but that's been on the block for 3 years now as well. So
yeah,
so it's,
I think this is really and I'm just joking, trying to create friction between you and Vincent and everything, because as a podcast host, I thrive on conflict. See what I mean? That's what it's worth. Okay. So Murali, I want to hear about your experience, and tell me again, similar question. How did the idea come to your mind? What idea, and how did you go around it? And then let's have Saad reflect to it.
Sure. My question was very simple. What is the best triplet in first relapse in multiple myeloma? That was a question which I wanted to answer. Straightforward question. This started approximately 3 to 4 years ago. Again, it's very similar to Hira's story, no different in the US version of the Hira story. That's what I would say. We started with this question. At that time, CAR-T trials were slightly showing promises. Bispecifics have not fully entered the clinical space, and that is where this is. Griffin, I think Saad is the best answer, but
So you do know most of my listeners have no idea what Griffin is, right?
Fair enough. Okay.
So you got to tell us a few of these things.
Sure. So in relapse, in first relapse of myeloma, there are multiple 3-drug options which are there. But the question was, most of these were done against 2 drugs, and we always know that 3 is better than 2, but never a direct 3 versus 3 in a randomized phase 3 in order to see which one will give the better clinical endpoints, either it's progression-free survival or overall survival. So this was a question in the field, and I still think this is a question in the field at this point of time. At that time, Daratumumab was the commonly used first relapse regimen, but we did not know which one would be a better partner. Is it Daratumumab with carfilzomib, or is it Daratumumab with pomalidomide? And this is the question, and we believe that these were the 2 best and less toxic first relapse regimens to answer. So this is where we started the idea, what is the best first relapse regimen? At that time, Daratumumab was just being started to be used in the frontline setting because the Griffin trial of 4 versus 3 had not read out. And we envisioned that even if it comes up, it will still be a relevant question to answer in the field. So started with ECOG, and it was Vincent, Shaji, and Sagar, Mike Thompson. So they were all there. We discussed the concept at length, and after about 2 to 3 months, we agreed that in ECOG and looking at other cooperative groups, including SWOG, that they were not doing a relapse trial and first relapse, we said, okay, fine, let's go ahead. So wrote the concept, and then ECOG executive committee approved it after approximately 4 months. And then after a couple of months, it went to NCI CTAP. But before that, we sought Janssen's approval. We got Janssen to buy in, and they said, yes, we will support the trial with from a drug standpoint. So we secured the funding, ECOG CTAP approved it, we had a valid question, and NCI CTAP. I presented the trial. The trial was powered for progression-free survival, but then it did not go through NCI CTAP because they had 2 valid concerns. One, power of first relapse trial for overall survival. And the second one is it was not innovative enough. So we are now back to the drawing board with the same question 3 years later, and trying to answer this question with carfilzomib, pomalidomide, dexamethasone, or carfilzomib bispecific antibody. And now it's been approximately a few months, and we are trying to get Janssen's support for this because we have to secure the funding first, even before it moves through the chain. We still think it will be a relevant question. But again, I think one of the difficulties as a young investigator I'm facing is the inertia. At which point, I'm always afraid, at which point will this concept be dead in the water? And again, I have to come back to the drawing board. That's my constant fear. After 2 years, if it gets rejected again, it has to start all over again. And this is despite being in a cooperative committee with Vincent, Shaji, who are all good myeloma clinicians as well.
This is about three years right now, almost 3 years.
Mm-hmm.
Yeah, Saad, I wanted to reflect as well, but I want you maybe as you reflect, it seems like there's a very similar experience, although different countries. But as a listener, I'm just putting my listener hat. there seem to be, to me, like three issues, and I want to reflect on those. One is the process itself. It takes months to years even to learn. Like, would have been nice for Murali to know that, you know what, the NCI wanted overall survival endpoint way before, it should not take 2.5 years for him to know that. There's one piece. Why is the process too prolonged? Number two, the funding mechanism, which Hira is still facing, but Murali was able to secure, and still, despite that, was there an issue with that? And number three is, really the way I would look at this is, how do I describe it without offending the academic cultures? It's very important for junior investigators to have some of these studies under their belt. We all know that, whether we like it or not. Sometimes promotions are dependent on this, recognition dependent on this. And, as much as I hate to say that in academia, having these trials and publications very important career-wise, but it is a fact. And I hate to see, 3, 4, 5 years going by thinking without having something fruitful. So reflect on these as you address these points.
No, I think these are genuine concerns, and I would be lying if I said it's not going to get harder. And the reason for that is, funding remains an issue, both in Canada and the US. The number of randomized phase 3 or phase 2 studies, depending on the patient population, that the NCI mechanism can support is only 2 or 3 in the frontline setting. Similar number in the relapse setting. And you have 3 very good cooperative groups, the Alliance, SWOG, and ECOG, with smart people who are trying to come up with ideas. If we can come up with good research questions, enroll patients together, and move on to the next trial if the mechanism was fast enough. But it's, I think part of the reason the system is set up this way is to perhaps, control the number of studies that can actually go through the system. So that's my observation and comment. The second important thing is the questions that both Hira and Murali and the ECOG, SWOG, all of us are trying to ask are non-pharma questions. They're academic questions, and bulk of the trials being done right now are registrational studies, how to get a drug or a combination approved in a certain setting. And they have really, taken over the bulk of what, the kind of trials that are being done right now. And they are innovative studies. A lot of innovation is coming from pharma. The academicians are not creating CARs and bispecifics or small molecules that we're trying to use for our patients. So, there is a disconnect. Those trials are reading out fast because, companies move fast, get sites in the US and ex-US and get the patients that they need, but we're, going through this process of developing a concept, submitting it, trying to get funding, and that whole process I feel is a bit, antiquated. And we've talked among ourselves. We have brought these issues up, as well, and we'll continue to champion for a better system. But I do feel that this will continue to be a long-term problem. I think one of the fears that I have is in a few years, we won't be the ones that are driving the research questions, in academia. Because I think a lot of this will be coming from the industry. We are seeing this brain drain to industry as well. A lot of smart investigators are leaving academia because of these frustrations and going to the pharma side of things. So if someone makes that choice, there's nothing wrong with that choice or motivation. But I'm just saying that, that also, lends to, this sense of, not getting that academic recognition or satisfaction that drives people to that decision making. So, So
Yeah.
those are just,
No,
some of my reflections, Shadi. I'm sorry, I
yeah,
don't
yeah.
have-
No, very, very important. Hira, are there other mechanisms to fund, for example, your trial except pharma? I mean, are you able to seek funding from somewhere else?
So for the type of question that I'm trying to ask, there isn't, because it's not, pharma's not interested, right? But I do appreciate Saad's comments a lot. And, I struggle with that sometimes, because it's hard to know how much energy, honestly, to put into a cooperative group trial that maybe is never going to move as a junior investigator, but also how to kind of get into the loop of some of the pharma trials, which, are, again, hard to break those barriers when you're just starting off. So for my particular trial, it's going to be the big Canadian funding agencies that we're going to be applying large grants for. And again, it is good to get some of the global support as well. So, we'll go in with strong letters of support from lots of international key opinion leaders, which will be helpful. But otherwise, I sometimes wonder about patient advocacy groups and so we're going to look at that as a potential mechanism for parts of our question, not the entire trial. But for example, for my particular trial, the cost of the drug is not an issue because we're going to use standard of drugs. And, if at some point the standard of care in Canada reimbursed were to change to a 4-drug instead of 3-drug, we would be able to adapt to that. But we can't, pharma's not particularly interested in a de-escalation type question. But, again, questions around trials which focus on quality of life and health economics, I sometimes wonder if there was an opportunity for some, our Ministry of Health to kind of come in and say, you know what, if I save a year of Daratumumab, I can fund the entire trial for the entire duration. But it's this disconnect, this red tape between all these different agencies where there's absolutely no crosstalk. So even though I actually work in a publicly funded national healthcare system, each agency has its own different mandate. And you can't see, if you save money here, you actually save money for the whole country, but it's just not seen that way. But again, those are some of the barriers and there are lots of, great examples of countries that do use more of where their government is much more supportive of cooperative group trials. And that's something I think that, at least in Canada, we really need to move towards.
Murali, do you have any opportunities to fund this outside of the maker of Daratumumab?
Not much, Charlie. And I agree with Saad's point, and I actually want to ask Saad. Saad, that's a great point which you made, that most of the trial, more like, for example, currently the Daratumumab-based trials in first relapse versus a bispecific is being run by pharma, which is very fair. The question then becomes, what will be the role of us as academic clinicians, and how can we then contribute? Because we are foreseeing a future now where Dara may be used or may not be used very well in the frontline setting, but there's enough evidence. What about these patients in first relapse if pharma just runs a Dara-based trial? What are their options, and how do you envision the academic's future or our role in this?
It's important, Murali, because that's a very good question, and I can share some of my thoughts because I took over from Paul as the chair of the Alliance Myeloma Committee in December. So the cooperative group issue is now actually a problem that I think about a lot as well. Yes, pharma is doing those randomized phase 3 studies, but what's the true number of patients from the United States institutions that are going to be enrolled? It's probably going to be less than 10%. One can still make a strong case about asking an academic question with our pharma colleagues in specific patient populations. We know that the practice in the United States, a lot of patients are going to be progressing on lenalidomide right after that first relapse. So, design a study that kind of addresses that particular question in that particular space. It's just as an example, to what you're trying to do, but make sure that you power it in such a way that you're actually able to get the question answered. It's okay to negotiate with CTEP or the NCI Myeloma Steering Committee. When you do get that feedback from them, there are going to be 6 or 7 or 8 points that they make. They're going to be flexible on most of those points. Try to identify the points that they will be, they will say, all right, if we can't negotiate on this, you have to make this change. If that change, or rather, if that specific, suggestion makes your study impractical in terms of reading out 10 years from now, it's okay, just move on. But if, that impossible, change is palatable and you still get your answers 5 or 6 years from now, proceed, with that idea. But my advice would be persevere. That's the life of a clinical investigator.
Saad, can I ask you a question that just comes to mind? So,
yes,
in your answer to my previous question, you said you've got Alliance, SWOG, and ECOG, and there seems to be some competition on resources between the cooperative groups because funding cannot be assured with this. Why do we need 2 cooperative groups? How about one cooperative group that gets these resources? I mean, why do we compete against each other?
So, United States is more like a continent rather than a country. It's a big geographic area. And, the cooperative group mechanism used to be, much more broader, say 10 years ago. I think there were like 15 or 16 different cooperative groups, and they were pared down to these 3. So the effort was exactly what you're saying, create, a more uniform way of doing things. And these cooperative groups were geographically divided, but there is, if you're a SWOG institution, there's no reason why you can't do an alliance trial. And that's kind of the deal that the 3, disease chairs made, that, if ECOG is going to propose a study, SWOG and Alliance will support it. If Alliance is coming up with an idea in the randomized phase 3 setting, the other 2 cooperative groups will support it. So that's how the SWOG 1803 is being supported by all the cooperative groups. There is no other maintenance trial right now in that setting. ECOG, when endurance trial was run, the KRD versus VRD, all the 3 cooperative groups, even though it was an ECOG idea, we were enrolling patients on those trials, and on that particular study. So, we've, what you're making a good point, but we've already kind of moved from that wild west to a more uniform.
That's very helpful. Murali and Hira, Murali mentioned this, and I want to have your reactions to this. Murali said, well, what is going to be the role of academicians? And Saad, you answered this, but if pharma is sponsoring these trials. So my question is to you, Murali, and to Hira, is there are some that might argue, well, just say no to pharma. If a pharma comes in to you with this pharma-sponsored trial that you think is useless and it's not helpful, just say no. And then you almost like forcing the hand of pharma to do the trials that you think as an academician are mostly important. So nobody is forcing you to open a trial that is pharma-sponsored at your institution. You can simply say no. And this way, if all of the investigators say no to the pharma studies that we don't think are helping patients or are academically inclined, then pharma will have to go back to the drawing board and must design better trials that align with the academic mission and with what investigators think. I'd like to have your reaction, the 3 of you, to this. I'll start with Hira.
So I guess my feedback would be that I think that a lot of times, most of the times, a lot of the trials that are being done by pharma are actually really innovative and are really helpful. They provide my patients with better options than standard of care. So for me, the ultimate line for not opening a trial at my site is only if I think my standard of care is better than the control arm. That's the only time I would say no. So, but you have to remember, I again practice in a different setting, right? So if a comparator arm, if a control arm for me in first relapse is DVD, that may not be your standard of care, but that's my standard of care. And so I will open that trial. If it's a doublet with KD, I will open that because that's my standard of care. But if I had a VD, for example, as a control arm, I would not open that trial. So for me, that's kind of the ultimate. Again, as Canadians, we're always kind of in the middle ground. We never debate. We're always neutral. I wish that there was just more opportunity for both, again, investigator-initiated studies with pharma where we can work together. And often, one of the challenges, again, I have, and Saad briefly, talked about this, but if you're trying to propose a trial with two different companies, it's like impossible, like forget it. And so then the only mechanism I have is through a cooperative group. So I wish that we could actually convince pharma to continue to open lots of trials, continue to do innovation. We want to be a part of it. And I wish that there was more of a mandate from pharma to say, we are going to open X number of investigator-initiated trials, X number with early investigators in it, and have us be more involved. But I honestly think it's a partnership, especially in Canada. I rely on pharma to bring therapies to my patients that I don't have access to. This may not be the controversial answer that you had wanted. I'm sorry.
No.
That's my reality. I don't have kind of any funding for academic trials at my site. So I rely on the surplus revenue generated from pharma trials to open, the SWOG trial, Saad, we were unable to open at my site. The maintenance trial because the deficit was just so high. I couldn't make it up with all the pharma trials. So I need the pharma trials. I just want more of a partnership and ability to, again, steer as a junior investigator some other questions.
Murali, can I make that comment?
Yeah, but I just don't worry. I'm not looking for controversial answers, just honest answers. Saad, go ahead.
So I want to get to that point. So the practical aspect of this issue is, we need money to fund our clinical trials research support infrastructure, right? So the cooperative group mechanism, is a losing proposition. It's a flat fee, very low, and you always lose money when you do an NCI CTEP run clinical trial. So, the institution has to have a balance of doing IITs and pharma studies, because that's where the resources come from. So I think it's a balancing act. I agree with Hira. The standard of care arm issue is a real one, and that's the reason why a lot of the pharma trials end up enrolling patients, in Europe, in Asia, and in I should say Australasia, because access to drug is not the same. Even for you, Hira, the access of drugs is better than a lot of places in the world, right? So in the US, we are super blessed from that standpoint.
Saad, sometimes these trials that are using a substandard control arm and are used with the FDA to approve the new drug, despite substandard control arm, it lends to approving a drug, and then the drug gets used in the US while we know that the control arm was substandard. How do you reconcile this? Because that trial was conducted elsewhere.
I agree. Yeah. No, I agree with that. I think, the time when the 3 versus 2 drug trials were being done, and we actually have some recent examples, but most of the big academic centers stopped accepting those trials. So the last 3 versus 2 drug study that I got involved with was Candor, and that study started and was designed back in late 2016. And at that time, we were just learning about the early data from Pollux and Castor, and so but since then, most academic centers, they don't accept those arms, because that's simply not our standard of care. There are investigators, however, who open those studies and participate in it for anyway.
So Murali, let me have you reflect on the first question. I want to go back to the control arm because I have a provocative question for all of you. I'd like you to react to. Just came to my mind, by the way. But Murali, do you feel that you have the power to kind of force the hand of pharma to say, okay, I'm not going to open these trials. So they get stuck. They must open a study that you as an academician agree to.
So I think when I open a study, the real question is, like Hira and both sides said, is does it benefit my population? That is my first thinking when I open a study. And if there is a chance that it benefits the patients whom I serve, that takes precedence over an academic question whenever I open a study. So that's the first thing, Shadi. For example, the Dara bispecific versus Dara POM dex or Dara Velcade dex. If the standard arm, if the SOC arm did not include Dara POM dex, it would be nearly impossible for us to enroll it in the US with the Dara Velcade dex alone. And the pharmaceutical companies know this. So that is why there are two options, and maybe Dara Velcade dex is not available in other parts of the world. And for that population, that might be appropriate. So I think every investigator, when they open a study at their center, thinks more of their population and access to that particular drug rather than the academic question. So that's what I want to say.
You know, it's interesting. I'm still flabbergasted by the years, 4 years now, Hira is going, and 3 years for Murali is going. And frankly, let's be honest, we still don't know whether any of these two trials will open. I mean, let's be realistic, right? And if they open by the time they enroll, and we're talking 6 years from now, will be, and who knows where the myeloma field will be there. But here's my question, and I just thought about this, and I'm actually really curious your thoughts, and I'll start with Hira. So we talk about the substandard control arm in a clinical trial setting, but shouldn't we be looking also at the real world uptake of what we think the proper control arm is? I'm just going to give you an example. So let's say in Canada, the proper control arm usage is KD or whatever. Like it's a reasonable control arm, right? KD, right? But theoretically, let's say we conduct a real-world data analysis of patients in Canada with that particular disease in that particular setting, and we find out that, you know what, 70% of these patients are not getting KD, they're getting D or whatever it is. Isn't it fair that maybe if you do a clinical trial that incorporates novel agent, then plus D against D will be still okay because you get access to this newer drug, and you're not really saying the patient's receiving substandard control arm, because in the real world, they're not getting what you think they should be getting. I guess the simple way to phrase my question, which has gotten too convoluted, is what we think is the control arm is what we believe patients should be receiving. But we know that patients in the real world don't always receive what we think they should receive. So is it fair to design a trial with the control arm being the drugs that are mostly used in a real world setting?
So
let's go.
Go ahead, Saad, because that's a hard one. So maybe you can go first.
You start.
Okay. All right. So let me, give a 30,000, foot view to this a bit. So that standard of care, Chadi, that keeps on evolving and changing based on regulatory approvals. So it's not a constant. If a trial is enrolling over a 3-year period, given the fact that we have so many other randomized studies that are reading out and companies getting to regulatory approval, those options being added to the quote unquote standard of care that's out there, your standard of care arm gets extremely heterogeneous, and you lose the power to answer the question that you started off with. The answer to that question is, if the standard of care was consistent, over a certain period of time, that's totally fine, because what you're doing there is your experimental arm is remaining the same over a course of 3 years of enrollment, but the standard of care arm keeps on changing. So you won't be able to you won't be able to predict what the measure of success is, or to achieve whatever that hypothetical measure of success would be. So that's my thought.
I mean, I guess the question is, Hira, is and Saad, what you said is very, very important. But is the control arm, should the control arm be what the control arm has been in prior clinical trials, or what patients indeed receive outside of trials in the real world?
I mean, but what patients receive outside in the real world may be different in different regions.
Right.
Shadi. So
So
you won't be able to control for that.
Yeah. I mean, but I guess what I see in the real world is not always consistent with what clinical trials has published or has suggested or has recommended. I think it is fair assessment that patients in the real world don't always receive what we have theorized to be the best therapy because of a variety of reasons. Access, lack of education, perception of poor tolerance, whatever it is. But if you right now, you go and look, for example, at real world data of whatever lung cancer or maybe myeloma, you are unlikely to see that consistency of even 3 versus 2. You'll see a lot of heterogeneous approach. And if you're designing clinical trial and you're bringing a novel agent to that trial and you're picking a control arm, do you choose a control arm based on what you are seeing patients receiving in the real world, or based on what has been established in prior clinical trials? I think it should be clinical trials, but I can understand when you actually choose real world data.
So if you have a very well-controlled experimental arm and a not controlled heterogeneous standard of care arm, the experimental arm will be positioned to succeed if it's an effective therapy, right? So in that case, my bias would be that you're already, actually favoring the experimental arm to kind of win in that scenario. The point that you're making is absolutely right. Do all the patients who start on, a carfilzomib-based triplet continue that regimen, for 5 or 6 years? No, you get to a certain, response, and then you drop off, 1 or 2 partners, and then just keep going with one. The same with a Dara triplet. You get to a certain response, and then you're trying to pair things off and make things better for your patient. So are we following those clinical trials, from a practical standard of care standpoint? No, I mean, we are taking care of patients, based off of, their decisions, their lifestyle choices, their safety profiles, etc. But will the outcomes be the same as clinical trials? Probably not. Maybe, who knows? But the assumption would be that the patients, in the grand scheme of things, in the full picture, if you were to look at that heterogeneous pattern, you won't get the same, kind of PFS or responses as things were done in the clinical trial. Does that answer your question?
Yeah, no, it does totally. I mean, it's just a question just came to mind because you see so much heterogeneity in how patients are treated. You've been very generous with your time, by the way. I want to be very sensitive to your time. I know on the East Coast it's getting a little bit longer. For listeners, we're taping literally on Saturday night because we don't really care. Saturday night, Friday night, we're always working. I'm just saying that. But I do want to finish with an important question that all of you want to react to, because it stems a little bit from the latest ASCO meeting that we actually had. Even at the time when a trial is finished and complete accrual and the results are out there and presented, whether it's plenary session or non-plenary session, there remains controversy into how to interpret the results. So not only it's taking like 3 years and 4 years and 5 years to even launch a trial, if it gets launched, and maybe 10 years to get published. But even if it's published here, people will probably not buy your conclusions. And the same for Murali. I mean, that's what's probably happened. The determination trial comes to mind. Again, I'm not a myeloma expert, but obviously I've listened to the presentation, I've listened to all of this. But this is an example where a trial took a long time to be published and presented, was a plenary presentation. And I think it's probably fair to say that there remains controversy into how to interpret the results. I sense that, maybe I'm wrong, but I sense some people said, well, this means I don't need to transplant. Some people say it means, no, I can still transplant, but maybe I don't need to twist the arm for a transplant, whatever it is. Tell me your reaction, Hira, about the determination trial, and what's your sense about how long it took to enroll, and whether you believe it created controversy, or do you think it solidified the role of transplant?
So a couple of points about this one. So, it's interesting because we always talk about best treatment up front. We always talk about attrition. We always talk about never making it to that second line of treatment when we talk about a lot of other effective regimens. But for something like transplant, even with the improved PFS, it didn't have the OS. Somehow those issues, I felt like of attrition and getting to that second transplant in the real world have been a little bit less emphasized. So, and some of it, again, I wonder about whether or not, if this was a really cool non-melphalan-based treatment, so if this was like a drug that made a lot of money, how would those results have been interpreted? Would they have been interpreted differently? In Canada, we are big believers in transplant because it's the absolute cheapest therapy that you can give to somebody. Exactly, Saad. So it's so for me, when I look at those results, I say, you know what? My very cheap, effective therapy that I can give in a publicly funded healthcare system, and I know my patients may not get to second line treatment for whatever number of reasons. Hey, this works. I got a good PFS out of it. It's better than my non-transplant arm. That's the interpretation I got to it, but it is incredible. I just sometimes, and I would love to hear others on their opinion. If this was truly a drug that had made a lot of money, would the interpretation of the trial have been different? Is it because transplant, is cheap and effective that there's so much controversy around it? I don't know. Again, those were just my thoughts, but I transplant. I will continue to transplant. This has further affirmed for me the role of transplantation.
Murali, how do you interpret the results and your reflections on it?
So I agree with Hira that if it was a drug which made a lot of money, we would have really been celebrating. With the respect to the determination trial, I think at least, so the first one is whether people have access to transplant in the relapse setting as well. If they have access to transplant for standard risk patients, for me, it allows them to completely choose at whether at first remission or at second remission or at first relapse. So that's how I am interpreting this, and also saying, look, you have a long PFS. I think it basically reaffirms that at standard risk, we can defer transplant provided patients will have the same reasonable access to transplant as they come and see me during the first time. Regarding the overall survival, I mean, I should say I support Saad's tweet, which is you have to always look at 10 years. Still, the follow-up is short enough, and we really don't know. I mean, even though the curves are very close, it might be very different. For example, at 7-year follow-up, if the non-transplant arm or deferred transplant arm already had, the patients had already underwent a CD38, carfilzomib, and pomalidomide, only now, later, then the curves might be separating, while if in the transplant arm, if it didn't, then the overall survival might be better. What is more intriguing to me is actually how did the PFS of the transplant arm disappear? So that's the most interesting question, and what equalized it. So those were my thoughts.
Saad, you did call this a very interesting name. So why don't you have your reflections on this and opinions on the trial?
So, again, I'm looking at this from the scope of having listened to talks from, many of my myeloma, colleagues who used to be like my icons, back in the mid 2000s. And I used to listen to these talks. Now that we have these novel agents, proteasome inhibitors and IMiDs, what is the real role of transplant? And, it's funny that, we're here in 2022, and I hear the same question, despite the fact that we've had several trials where high-dose melphalan, has shown us, these results. So first off, this is not an early versus late transplant study. It's early versus no transplant. That's what's been reported, because very few patients actually got a transplant, at this follow-up of time. What was the primary endpoint? It was PFS. If this was Maya being presented, we would be celebrating Maya, and that's what we did. Maya was an ASH LBA and reported very similar kind of PFS benefit. The OS issue, I agree, Murali, I try to point this out, back when the OS difference was shown, for myeloma in transplant versus standard of care studies, the OS for myeloma was 2 to 3 years. So you would see those separation of curves, and you'd be able to tell the difference, at the 3 or 4 year follow-up mark. But you can't see that now. Even for the len maintenance OS benefit, we had to wait for at least 8 years. And so, when you only have a shorter follow-up, and the OS is not met in both the arms of the study, the right conclusion is OS has not been met for both arms of the study. The statement that there is no difference in OS is completely wrong. Okay, so I kind of disagreed with my esteemed colleague when he presented the data and made that statement. But that's also some of the inherent biases that we have as investigators. If we design a study, we want to answer a study, the answer comes out the total opposite of what we wanted. We still want to, hold on to some of those biases, and what we need to do as investigators is do a bit better. So I hope, Shadi, this is enough controversy for you.
No, I love it. And you said it, the name.
So what's the final answer, Saad? Yes or no?
Daratumumab 1.
Yeah, Daratumumab. So quick question, but Murali's trial was returned back because his primary endpoint was not overall survival in the relapse setting. Are you saying in the relapse setting it must be overall survival? In frontline setting is progression-free survival, Saad?
So my short answer is, it should be PFS, for each the frontline as well as the first, first relapse setting. By the time you're waiting for OS, just like you highlighted, you've waited enough time. There are other registrational studies that have led to approvals, and now the standard of care has changed and the practice is changing. And your question, the answer that you try to answer with OS is no longer relevant. So I think that becomes a challenge when you have a disease that has an median OS of 8 to 10 years now. If this was PANC, it would be OS would be a reasonable, question.
But Murali, didn't they return your trial said because there was no OS primary endpoint? Did I miss that? Didn't you say that?
Yes.
So no, I agree with that. But what I'm trying to say is, that was the wrong decision, and that was not the myeloma investigators.
Yeah, no, that was the NCI. Yeah. Okay, well, look, we need to finish with concluding statements by each one of you. I'm going to tell you my concluding statement because you may laugh at this. When I did my internship, I'm going to tell you how we treated multiple myeloma. The median survival, by the way, was about 3 years. The way we treated myeloma, all transplant ineligible patients received melphalan and prednisone, and transplant eligible patients received VAD, followed by transplant. That was it, actually. That was how we treated multiple myeloma. It was easy for me to get that, just to give you an idea. So concluding statements, Hira.
So I guess, I have learned a lot during this podcast. Good to know that some of the challenges I face are actually not so unique, and they're actually, I feel bad. Murali sounds like he was way farther ahead and had to come all the way back down, but I appreciate Saad's comments around just persevering and just keep going at it. And I hope that, again, a lot of senior investigators like Saad, like Vincent, have really done a lot for junior investigators in terms of mentorship. And again, to emphasize that point again, that is absolute key, because otherwise I feel like we would just, I don't know, Murali, how you feel. Sometimes I feel like I would just get crushed and become so little. But mentorship through ASH CRTI, this year I'm doing the ASCO clinical trials course. Those are so key in seeing for junior investigators where we may be right and also where we may be wrong and how to improve on things. So that would kind of be my key point. I really appreciate it. Again, Murali and I have had similar experiences, so good to know, and really appreciate Saad's thoughtful comments as a senior investigator.
Murali, concluding remarks from your standpoint.
Yes, certainly doing clinical trials is extremely hard, and actually Saad sounds very much like Vincent now, in the sense that negotiate, accept, don't budge on certain things, but budge on certain things. So
Murali, I'm trying to get people to disagree with each other. I want Rafael Fonseca, Vincent, Saad, Sagar, they all like screaming at each other. That's how I get clicks on the podcast. You're making it so easy. Okay, fine. That's fine. You keep going.
Fair enough, Shari. I think I'm past a certain age. I think we all acquire wisdom. Sorry, I'm not saying you're old, okay?
No, I know. I'm older than that, by the way.
So I think Hira's struggles and my struggles is what Saad also came through. That's what I hear. I wish the system is better, or we can redefine academic purpose of our cooperative groups. Maybe I don't know. Maybe we should be focusing on not phase 3s or phase 2s or more translational questions if it's very difficult to fund. That's my thinking. Should cooperative groups still really do phase 3 trials? Just as a challenging question to do, but I will certainly persevere and try and answer this question. And it was great hearing Hira's thoughts. And it's a completely different public funded system, much more challenges than mine.
Saad, concluding remarks.
No, my final thoughts are, have already been echoed by both Hira and Murali. Persevere as an investigator. That's what we do. We try to make things better for our patients. The environment is not, very easy, but it's not inhabitable. So, just persevere, find like-minded people, and keep on asking, the right questions. Modify your questions, seek advice, and find your base group that continues to help and support you. And it will be a mix of peers as well as senior investigators, but that's how you get through this. This is not easy, and the landscape will continue to change.
Yeah, well, look, the three of you, you've been absolutely wonderful to have on the podcast. This has been the highlight of my week. I usually at the end of each podcast, I put a quote or something I really inspiring from the podcast, and you listen to it. But I just thought I'll let you know one of my favorite quotes is by Pelé, who is a soccer player, as you know, from Brazil, and he once said, "Success is no accident. It is hard work, perseverance, learning, studying, sacrifice, and most of all, love of what you are doing or learning to do. So there's zero doubt in my mind that Murali and Hira are going to be absolutely amazing. They're going to continue to be excellent investigators, and I'll be cheering for both of you on the sidelines. And Saad will provide all of the support along with many of the senior investigators. So thank you so much for coming on the podcast.
Thank you so much. Really appreciate it. And thank you for the t-shirts.
Thanks for having. Yeah, I'm gonna go for a run with wearing this now.
The best in business, I'm telling you.